diagnostic criteria, and also look at a few aspects of genotype-phenotype correlations with NF2 mutations, and some of the more recent events around NF2 variant classification and genetic screening strategies. So first of all, just to summarize the data in the most recent CB Trust report, that meningiomas account for around 42% of all primary central nervous system tumours in adults. And that seems to have been creeping up gradually over the last sort of 10 to 15 years, from about 30 to 35% up to 42%. Whereas nerve sheath tumours, which account for around 8% of all primary CNS tumours in adults, that seems to have been a bit more stable in terms of numbers. And the majority of the nerve sheath tumours are vestibular schwannomas. So both of those tumour types are much more common in adults than children, as I think with most tumours. And they're normally sporadic solitary tumours, but they are both the two main types of tumours that you would find in NF2-related schwannomatosis. So the schwannomatoses are related conditions, and NF2 schwannomatosis is the most common. And in our most recent UK data, based on partly just England, but also the UK as a whole, so looking at around 56 to 60 million people, the most recent incidence we found is around 1 in 60,000 for NF2 schwannomatosis, whereas LZTR1 schwannomatosis is around 1 in 527,000 people, and SMARTB1 schwannomatosis is around 1 in 1.1 million. So they're much less common than NF2 schwannomatosis. And then there are these couple of other genes that have been associated with schwannomatosis, but in far fewer cases, so only 1 or 2 cases reported for each one. So I just wanted to pick up on some of the historical confusion between NF2 and NF1. So obviously NF1 is more common, and the primary tumour that you would see there is the neurofibroma. But people also get things like café au lait macules. And I think that with the lack of imaging or genetic studies, historically there has been some confusion about whether or not NF2 was a form of neurofibromatosis, because quite a large proportion of people with NF2 schwannomatosis present with cutaneous tumours and café au lait macules, or they might have café au lait macules, but not as many as you might see in NF1. So there has been some historical confusion. But only around 1 or 2% of people with NF2 schwannomatosis actually have more than 6 café au lait macules. And you will probably have seen, if you've looked at the updated NF1 criteria, it does specifically state more than 6, and it's very unusual to find that in NF2 schwannomatosis. And also from the NF2 schwannomatosis criteria updates, the neurofibroma tumour has been removed from the criteria, so they're generally not found in NF2 schwannomatosis. So the take-home message from this particular slide is that NF2 schwannomatosis is not a form of neurofibromatosis, it's a form of schwannomatosis. So that's why they've been regrouped. And so just looking at the updated, just click that, just looking at the updated diagnostic criteria for a minute. The major criteria obviously is the bilateral vestibular schwannomas, which are found in almost everyone over their lifetime. They tend to develop bilateral vestibular schwannomas. So that's the major thing that's kind of a standalone criterion. You can also have 2 anatomically distinct relevant tumour types like meningioma or schwannoma, as long as they both have a pathogenic NF2, like an identical pathogenic NF2 variant. So they're sort of standalone features. But if you don't have those, then you can build up a diagnosis based on either 2 major or 1 major and 2 minor criteria. So a unilateral vestibular schwannoma, obviously they're quite common in the general population, relatively so, but they are a single major criterion. And then if you have that plus either a first-degree relatives, multiple meningiomas, or a pathogenic NF2 variant in blood or another unaffected tissue, you can have a clinical diagnostic or a clinical diagnosis. Or you can have one of those plus something else like a single ependymoma plus a single meningioma, or some combination of those. And more than one meningioma, more than one meningioma you can't have as a minor criterion because it becomes a major criteria, but you can have more than one ependymoma, and that would be 2 minor criteria. And you can also have more than one non-vestibular schwannoma, but at least one of those has to be a cutaneous schwannoma if the unilateral vestibular schwannoma is the major criterion, just because that reduces some of the confusion between the other forms of schwannomatosis, which I'll mention in a second. And you can also build this up with the eye features like cataracts or retinal hematoma or epiretinal membrane, but you can only count each of those things once. So if you get one in each eye, it's still only one criterion. If we look at the non-NF2-related schwannomatoses, with these in the updated criteria, essentially you have to have a known pathogenic variant to get the proper diagnosis. So with the NF2 criteria, it is possible that you can have a diagnosis of NF2-related schwannomatosis without having a pathogenic variant or a known variant. But if you want to get to a distinct diagnosis with SMARTB1 or LZTR1, you have to have the known variant. And that can be either in blood with a relevant tumour, or it can be 2 separate tumours with identical variants. But those variants have to be there for that diagnosis. And if you don't find them, then you would have, if it hasn't been tested, if the patient hasn't been tested, they can have a diagnosis of schwannomatosis not otherwise specified if they don't meet NF2 criteria. And if they haven't been genetically tested, they can be schwannomatosis not elsewhere classified. But for the non-NF2 schwannomatoses with a known gene, that has to be there to meet the criteria. So this is just to show how the genes are linked together, as well as the overlapping diagnostic criteria for clinical symptoms. Because the genes are quite close together in the genome, they're all within 9 megabases on chromosome 22. It seems likely that there is quite a strong mechanistic link there as well. And this is just to illustrate that the current hypotheses are a standard Knudson's 2-hit hypothesis for NF2 schwannomatosis, where you get a single variant affecting the NF2 gene, followed by, so that's in blood, that's your first hit. And then the second hit would be a somatic second hit in tumour, which would be the loss of the trans allele. So you've effectively knocked out all of NF2, so you've got biallelic inactivation. Whereas if you look at germline SMARTB1 or LZTR1 variants, then you have your first hit in either SMARTB1 or LZTR1, followed by, in tumour, the second hit that knocks out the entire chromosome 22q. But then you also normally would see a second somatic hit knocking out the other copy of NF2. So you basically always have biallelic inactivation of NF2, but in SMARTB1 or LZTR1 related schwannomatosis, you have these first initial hits in a second gene. So I thought it would be good to go over some of the overlap between NF2 schwannomatosis and other forms of schwannomatosis. Firstly, SMARTB1 schwannomatosis, and here the main overlap is obviously going to be multiple non-vestibular schwannomas. So without that genetic testing, it might be quite difficult to tell the difference between something associated with SMARTB1 or perhaps a mosaic NF2 schwannomatosis, where you wouldn't necessarily get vestibular schwannomas. And so since the association of SMARTB1 with schwannomatosis in 2007, several studies have obviously then jumped on board and checked all of their patients and found that overall you expect to find SMARTB1 variants in around half of familial cases of schwannomatosis that have already been excluded for NF2 variants, but then only 10% of sporadic patients that are not NF2 related. And there are much more sporadic cases than familial cases. So this overall is a relatively small proportion of the non-NF2 schwannomatosis. It does, however, have a potential to have meningiomas in around 5% of cases. So if you had a meningioma, possibly more than one meningioma, and a non-vestibular schwannoma, the criteria then do get a bit blurred again unless you have that genetic diagnosis. And it's also complicated by the fact that meningiomas have been seen as the presenting symptom, or in fact the only symptom associated with a germline SMARTB1 variant, although it's very rare. It's an occasional misdiagnosis there. So then looking at LZTR1 schwannomatosis, which was identified more recently, and again, LZTR1 is found on chromosome 22q, about 3 megabases upstream of SMARTB1. And so this was identified by Piotrowski et al. In 2014, when they looked at a series of patients with schwannomatosis where they'd already looked at NF2 and SMARTB1, and they'd selected people who didn't have a known germline variant, but did have known somatic loss of chromosome 22 in the tumor. So they knew it was 22q related. And then they found LZTR1 in around 80% of their cohort. So then we looked at our cohort, which was larger, but unselected for 22q loss in the tumors, and included both familial and sporadic cases. And we found it was around 26% in ours. And then overall, when other people have also tested their patients, it seems that LZTR1 will account for around 38% of the familial cases and 22% of sporadic cases of non-NF2 related schwannomatosis. So that's about twice as many, as you could see from my earlier slide looking at the prevalence. It's about twice as many of the sporadic cases, which are the majority of schwannomatosis cases. So then we also looked at a cohort of people who had a unilateral vestibular schwannoma plus at least one other schwannoma, so maybe more, but at least one. And we found that in 39 people, 3 of those had a germline LZTR1 variant. So this then led to this additional overlap of, in LZTR1 schwannomatosis, you can have a unilateral vestibular schwannoma. So we then expanded this study and looked at more people to see how prevalent this was. So there was a larger cohort of people who presented initially with a unilateral vestibular schwannoma plus at least 2 non-intradermal schwannomas. So the intradermal schwannomas tend to be pathognomonic for NF2, but these ones were not vestibular, but also not intradermal. And 20 of those subsequently went on to develop bilateral vestibular schwannomas. So they then met NF2 criteria, and they tended to have variants that were identified in blood. So 12 of the 20 had a heterozygous variant that was identified in blood on standard testing. And then there were 2 extra people who were found to have mosaic NF2 at a much lower level, which was screened using a higher read depth NGS panel. And then another 2 who had 2 identical variants in tumors, but it was not visible in blood, so very low level of mosaicism. And that means that overall, most of the people who developed bilateral vestibular schwannomas had proven NF2 schwannomatosis, whereas the other 4 remain presumed because they meet clinical criteria, but there was no known variant identified. And that may have just been there weren't enough tumors to test, and if there were more tumors, they might have got a proven diagnosis. Of the 50 people who didn't go on to develop bilateral vestibular schwannomas, 5 of those actually did have the germline LZTR1 variant, and 5, so the same number, had a non-mosaic NF2 variant in blood, but none of them had a SMARTB1 variant. I think, as far as I know, there still haven't been any proven SMARTB1 variants associated with a reported case of vestibular schwannoma. So this means that in this category, it's just as likely that someone would have LZTR1 related schwannomatosis as a non-mosaic NF2 related schwannomatosis. And this is sort of to highlight the importance of testing for LZTR1 in those cases. So the example I have below is a case that was seen before LZTR1 was identified as associated. And so there was this one person who presented with a unilateral vestibular schwannoma plus 2 other schwannomas at the age of 39 and was tested for NF2 variants, and there was no variant in blood. And they only had 1 tumor available, but loss of heterozygosity was seen in conjunction with a single nucleotide variant. So this was assumed to be, if they were mosaic, this would be the variant that they had. And so they tested, there was pre-symptomatic testing of his son, and in blood there was no variant. So they said, okay, you're not affected and you're not predisposed. But then later on, after LZTR1 was discovered, they were both retested for LZTR1 and they both carried the same variant. And his son actually went on to develop a spinal schwannoma at the age of 43. So it's one of the reasons why you don't get a proper diagnosis if you only have what you have a relevant tumor and you have maybe found NF2 variants or like biallelic variants, but because you can't be sure that that's really the cause, you can't really get a definitive diagnosis that way. Okay, so I'll just move on to some genetic risks associated with germline pathogenic NF2 variants. And this is a bit of old data on the right there, but I'll just go through this, that obviously NF2 variants are found as heterozygous variants in the majority of non-mosaic NF2 schwannomatosis patients, but they are also the most common variants found somatically in sporadic schwannomas and sporadic meningiomas. And so this data on the right is just some old data showing the difference in risk depending on whether you have a germline predisposition variant in terms of the age at onset. So if you have a sporadic vestibular schwannoma, they tend to occur most likely after the age of 40, between sort of 40 and 70, whereas the peak age of onset for NF2-related vestibular schwannomas is much earlier, between sort of 10 and 40, with a peak in the sort of 20s. And obviously, if you have a predisposition variant in NF2, then you have almost a 100% chance of developing bilateral vestibular schwannomas during your lifetime. So if we look at the data from our NF2 database from Manchester, we had about 784 families on the database where there was a known pathogenic germline variant, and the majority of these would be single nucleotide variants. So you can see from the diagram that most of them are nonsense and frameshift variants, with a smaller number of splice variants and missense variants. And then this sort of 20% here are the larger deletions and insertions. So this is to summarize a genotype-phenotype correlation study that was done quite a while ago now, but it's looking at people with known single nucleotide variants in NF2 and mapping those to their locations within the gene, in the gene, and correlating that with protein domains, and then looking at what is the likelihood of developing a meningioma if you have that particular variant in terms of the variant type and the variant location. So here we go. This chart here, you can see that there is a difference. If you group the variants by the firm domains, so this 5' region with 3 different firm domains has a higher likelihood of developing a meningioma than somebody who has a variant in the more 3' area, the globular region, or in the alpha helical region. And I think it's probably clearer on the bar graph that you can see there is a trend towards a lower likelihood of developing a meningioma the later on in the gene. And you can see that there are a few different peaks along the way, which tend to correlate with the junctions between each of these protein domains. And because meningiomas are used as a feature of, or a measure of severity of the disease, this gives you an indication that the variant's occurring towards the beginning of the gene, and also by type well, we're doing the all variants now, but they're more likely to develop a meningioma, and it's an indication they have a more severe disease. The next graph is the one that has just the truncating variants. And you can see that on the plot here, it's a lot more overlapping and a lot less clear, apart from the variants in the last two exons, 14 and 15. But I think if you look at the bar graph, you can see that there is still some variation and a general trend towards a decrease as you go towards the 5' end, the 3' end rather, of getting a meningioma. And there are still these elements of peaks at junction regions, which suggests that although these are labeled as truncating and they're predicted to lead to no protein, that there must be some variation there, that some of them are still more severe than others, and that's probably to do with maybe some of them look like truncating variants, but they turn out to be a splicing variant, or and perhaps a splicing variant that's in frame and therefore less severe. So this kind of data was used to produce this genetic severity scoring groups. So Dorothy Halliday et al. Made this system. It's a bit of a complicated table. I've actually merged two tables from the paper to show the differences with tissue mosaics, which are the only ones that are found in category 1. But then categories 2, 3, and 4 are mild, moderate, and severe phenotypes. And so this is also based on the type of variant that you see and also the location. And I thought it would be a bit clearer if I move on to the next slide to just look at the types of variants that I was comparing to in the previous slides, looking at the type and location and the on the bar graphs, to show in different colors that if you have any missense variant or any small indel in any exon, they're all considered to be mild. So wherever they are in terms of location, they're considered to be a mild phenotype. Whereas if you have a splicing variant, they're considered to be moderate severity if they are in the first 7 exons, but a mild phenotype if you have them in the 8 to 15. And then with truncating variants, they're severe if they are within exons 2 to 13, but only moderate if they're found in exon 1 or 14 and 15. I think 1 is missing from there, but 1 or 14 and 15. But because there is within there quite a bit of variation, I thought I'd go through a couple of things to show that these variants don't always behave as you might expect them to. So this variant, NF2-885 plus 1G2A, so it looks like it's in a canonical splice region, and the category on the genetic severity scoring system would class that as a classic mild phenotype. But in fact, this patient didn't present to the clinic until they were aged 79, and that was with a foot drop. And it seemed like their medical history suggested their age of onset was around 74 years of age. So it was a very delayed phenotype. And when they had imaging, they could see some small bilateral vestibular schwannomas and some other cranial nerve schwannomas. But apart from some mild hearing loss, they didn't seem to be particularly symptomatic. And the most striking feature was that this person did have some large spinal ependymomas and some other small schwannomas. And spinal ependymomas are only really seen in about 25 to 30% of people. So having multiple large ependymomas seems like it must be quite a specific association with certain types of variants. And so his son, 2 sons, also carried the same variant, and both of them were clinically asymptomatic at the ages of 40 and 41. And they both carry the variant, and they did both undergo imaging, and only one of them had any tumours. And again, it was a large ependymoma. So there seems to be an ependymoma phenotype associated with this variant. But apart from that, it's a very mild, delayed onset phenotype. And so the genetic testing showed that this leads to an in-frame deletion of exon 9. So you can see on the trace that it becomes overlapping sequencing peaks. And on the gene, you can see the location in exon 9. And this in-frame deletion means that probably most of the protein is intact, and that might be why it's not quite so severe. But the large ependymoma phenotype, it's unclear why that would be. So looking at missense variants, there are quite a lot of variants that have been reported. And Katie Sadler, who's a former PhD student in my lab, she did a study looking at finding all of the variants that have been associated with an NF2 schwannomatosis phenotype in the Manchester database and in UAB in Alabama. So they were known clinically reported variants. And she also looked at things like ClinVar online databases and also searched the literature for as many unique variants as possible. And then compared the variants that have been associated with a schwannomatosis phenotype with those that had not. So looking on places like Nomad. So in total, there were 395 variants that she found, and 97 of them had been associated with a schwannomatosis phenotype. And she firstly mapped those to which exon they occurred in, and then looked at the frequency of their occurrence per exon. And also she looked at the size of the exon, so it was sort of a per nucleotide frequency for each exon, to see which ones were most likely to have missense variants. And from that data, it looks like exon 7 is the most likely to have a variant in NF2. And if you look at the bottom, the pale blue line, that's the Nomad variant rate. So that's people who are not known to have NF2, and it's fairly flat. And then if you start including the variants that have been associated with the phenotype, you can see it's much more changeable. And so exon 7 is the most likely, but also exon 2 and 11 look like they're quite strong contenders as well. So she then looked at each of those variants that have been associated with a phenotype and reclassified them according to the current ACMG and ACGS guidelines. So all the variants had been obtained from data that was spanning back maybe 30 years, and a lot of those would have been associated with the phenotype before the guidelines were put into place. So she wanted to standardize all of that data and then map them again onto the gene. So you can see in this image of the NF2 gene with all of the little lollipops representing the variants. Anything with a grey stick is something that was found on Nomad or in a database not associated with NF2. And then the ones with a red stick were the ones associated with NF2. And when she reclassified them and mapped them all onto the gene, you can see the ones that are sticking up at the top with probably too small to see the labels, but there are only 8 variants that were still considered to be pathogenic or likely pathogenic after the reclassification. And there doesn't seem to be a specific domain where they cluster. And so it's difficult to tell whether, like, there was no particular domain that stands out as, or maybe they all cluster here, and if it's somewhere else, then maybe they're not pathogenic. But I'll come back to one of these a bit later on. So if I start looking at the genetic testing strategies that are used to find these variants, we've had a look at, again in Manchester, at our data of second families who have NF2-related schwannomatosis. So we're ensuring that they are non-mosaic by making them second generation. And these are people who've all been tested by the standard panel of an NGS panel, including NF2, which also has SMARTB1 and LZTR1 on it, and also MLPA analysis for copy number variants. And it's similar to what you might expect, that the the most common type of variant is, if you combine them together, nonsense frame shift variants, which is the blue and, so it's the orange and grey together. And then the splice variants are quite common and probably more variable. And then there are fewer missense variants and only one person who had an in-frame deletion. But overall, in that cohort, using the standard pipeline for testing people using NGS and MLPA, there was an overall pickup rate of 87% of people with a known, an identified variant. So for the people who didn't have an identified variant, we then wanted to start looking at what else can we do to find a variant. So we're pretty sure they have a diagnosis. They're in a family that has the right phenotype. What else can we do to find the variants? And some of those can be found using karyotyping if there's a chromosomal translocation. But we've also used things like in silico analysis, genome sequencing, and a cDNA analysis to either find or confirm variants. And so I just wanted to highlight a few examples of that. And the first one is using in silico analysis. So there were two families where on screening, we could see this -66 to 65 insertion of a T variant in the untranslated region. But you can't really classify that. It wasn't doing anything particularly obvious because it's in a non-coding region. But then Nikki Whiffin, who you might have heard of, contacted us to ask whether we had any variants in the 5' UTR because she was developing this in silico analysis pipeline to assess this type of variant. And so she put this into her pipeline and showed that, first of all, I guess I'll explain, in the wild type situation, it's known that in the 5' UTR of the NF2 gene, there's an upstream open reading frame. So there's a start site before the official canonical start site in exon 1. And that's thought to produce a relatively small peptide with this stop codon, which is upstream of exon 1. So that doesn't interfere with anything to do with the coding, the expression of the NF1 gene. However, when you introduce this single nucleotide variant, it does two things. One is it looks like it creates a new start site, although the peptide that will be produced is only about 2 or 3 nucleotides long, and then there's a stop, which is still upstream of the canonical start site. However, it also changes the reading frame of the pre-existing upstream open reading frame, and that causes this transcript to start or continue reading through into exon 1, which would then interfere with the normal expression of NF2, which starts from the canonical start site within exon 1. So this has been found to be causative in 2 different families, which would previously not have been identified before this method of testing would have been introduced. So then also we've done some cDNA analysis to test the RNA for splice variants. And this variant is one where you may remember earlier I talked about a C885 plus 1 G2A, which had a very mild phenotype associated with ependymomas. And that was found in the canonical donor site, whereas, I go back. This is found outside of the donor site, but still within the splice donor region, which goes from the -3 position within exon 9 up to +6 here. And it was predicted by Splice AI to have an effect on splicing. And when we did cDNA analysis, you can see that there was indeed an effect on splicing. And it's exactly the same type of change that we saw in the 885 plus 1 G2A. And the person with this variant also had ependymomas. So it does seem, I think there is another person who has been seen with the same variant, so maybe 3 altogether, with the same kind of donor region variants who all have ependymomas. So it does seem like there's maybe a correlation with that type of event and the appearance of ependymomas. So then another variant, which is even deeper into the intron, so it's outside of the splicing region altogether, but Splice AI still predicts that it does have an effect on splicing and causes a donor gain, which should be stronger than it weakens the canonical donor site. And splicing analysis on this does show that there is an effect, and it weakens the canonical donor site and causes the insertion of those the first 56 nucleotides of intron 1. And so that's leads to a truncating effect because there's an in-frame stop codon within the intron if you continue reading through. And because this is exon 1, you might have expected it to be relatively mild because it's what's written in the genetic severity scoring system. But because it's after the end of exon 1, it kind of falls outside of that region. And the people who have this variant actually do have quite severe NF2 with an early age of onset and the appearance of bilateral vestibular schwannomas at an early age, and at least one of them has meningiomas as well. So this is more of a severe phenotype associated with an early truncating variant. And the final splicing variant that I'm going to show you today is this 1532 A to G variant, which you probably won't have been able to see in the plot that I showed earlier, but it's one of the missense variants that was considered to be potentially pathogenic on the missense variant review. And if you have a look at the Revel score for its potential effect as a missense variant, it's relatively high, so might gain evidence for pathogenicity based on in silico analysis, just looking at its effect as a missense variant. However, it also has an extremely strong donor gain prediction in Splice AI. So we did RNA analysis on this as well and showed that this also has an effect on splicing and actually produces two different aberrant truncating variants. So they're both truncating, and this is also leading to pathogenicity. So it's one of the reasons why I think there might be some variants that don't really conform very well to the genetic severity scoring system, because they might be doing something that you don't expect them to be doing based on just looking at them in silico. And so finally, another type of variant that we looked at for somebody who didn't have a pathogenic variant identified on next-gen sequencing on the panel and MLPA, they were referred for genome sequencing through the UK's 100,000 genome sequencing project. And looking at that data with the standard pipeline that they use, no variant was identified there either. But one of the clinical scientists here in Manchester, George Burgle, got hold of the data and had a look and did some manual reanalysis. And he found evidence of a large rearrangement, so a complex rearrangement that spans around 15 megabases on chromosome 22q. And so that gives a diagnosis for the patients, but also Christina Perez-Becerril, who's a former postdoc in my lab, has decided to continue looking at that variant and do some extra long read sequencing. So it's whole genome long read sequencing, looking at the same, but focusing on looking at the same region. And that confirmed the breakpoints that were found on the short read whole genome sequencing, but also identified a couple of extra possible breakpoints, which we're still working on characterizing. But this kind of long read data has the potential to find more variants and also give more information in terms of, if it's, say, a complex rearrangement, a bit more information on the organization and directionality. So just to summarize that, the additional experimental analysis that we did on people who didn't have an identified NF2 variant on standard testing meant that we could increase the detection rate to around 95%. And this means that for a lot of labs who have historically found somebody didn't have a particular variant, that it would be worth reanalyzing some of those for things that are non-coding, so untranslated regions and intronic regions. And also, as all of these new technologies like long-read sequencing are introduced, it can mean that we can identify more variants and also look at characterizing those variants in more detail, which will hopefully give a better idea of not just diagnosis, but prognosis as well for the people who have the variants and whether they're more likely to be mild or severe variants. So I think I think that's the last slide. I'll stop there. And this is just a list of the people who I've collaborated with on some of those studies and the funding that's supported some of those studies as well. But that's everything I have for today. So thank you.